Now that we've had a chance to go through mitosis and meiosis, let's do a little bit of comparison contrasting between these two processes. So remember, in mitosis, the whole goal is to create genetically identical daughter cells that are the same as each other and also identical genetically to the original parent cell. So if you start out with, for example, in humans, a diploid parent cell, which has 2 sets of chromosomes, you will end up producing genetically identical diploid daughter cells, again, both having 2 sets of chromosomes. Meiosis, things are a little bit different. Right here, you have to start out with a diploid parent cell, and you end up dividing the sets of chromosomes, one for each daughter cell that results from the cell division. And so the resulting daughter cells are only going to have then half the number of chromosomes as the original diploid parent, and the daughter cells will be haploid. On top of that, mitosis produces 2 daughter cells, but in meiosis, because we go through 2 rounds of cell division, we end up creating 4 haploid daughter cells. This is a really good figure from your textbooks that allows you to follow the exact same diploid parent cell through either the process of mitosis or the process of meiosis to see how the 2 processes differ. Right? So in both cases, this parent cell will have to replicate its DNA before it goes either into mitosis or into meiosis. You'll notice that in mitosis, all the chromosomes, duplicated chromosomes, while homologous chromosomes exist in the same cell, right? So this guy is homologous to that guy. Here are the 2 medium-sized ones, and there are the 2 short ones. Each of the homologs pays no attention to the other partner. And so all of the chromosomes, the duplicated chromosomes, will line up by themselves on the metaphase plate, right? So here are the 2 long ones, 2 medium-sized ones, and the 2 short ones. In meiosis 1, this is very different, right? In prophase 1, you've got synapses forming or taking place to form the tetrads. This is also where crossing over happens, only in prophase 1. And because of the crossover, because of the synapsis, those homologous pairs are matched up really tightly with each other. And so as the chromosomes line up in metaphase 1, they have to line up together as pairs at the metaphase plate. So compare metaphase 1 here to metaphase of mitosis. That's a very different organization of the chromosomes. Because the homologous pairs are lined up together at metaphase 1 of meiosis 1, you end up separating the homologs here at the end of meiosis 1 during anaphase. So here you can see each daughter cell now gets 1 set of chromosomes, 1 long, 1 medium, and 1 short. These cells are now haploid. The trouble is, though, that their chromosomes are still duplicated. That's too much genetic information, so the cells need to go through 1 more round of division to unduplicate the chromosomes. Remember that meiosis 2 is identical to mitosis. So in this case, each individual duplicated chromosome will line up by itself at the metaphase plate, just like what you see here in mitosis, separating the sister chromatids, so that you end up with 4 daughter cells, each with only half the number of chromosomes the original parent cell had. Right, so the original parent cell here had 6. Each of those daughter cells now only has 3 chromosomes. So they're all haploid. They're also all genetically different from each other as a result of crossing over and the assortment of chromosomes at the metaphase plate. This particular slide I stuck in there for you guys to use as you're studying. It's pretty much text that summarizes some similarities and differences between mitosis and meiosis for you to look over as you're studying. All right, so let's review. There are 3 events that are unique to meiosis, and they all happen in meiosis 1, because remember, meiosis 2 is the same as mitosis. So here are the 3 things to remember from meiosis 1. First of all, in prophase 1, this is where synapsis and crossing over take place, the only place where they take place. Right? So crossing over allows the non-sister chromatids to exchange some of the DNA, mixing mom and dad's DNA onto the same chromosome. In metaphase 1, this is where the homologous pairs of chromosomes line up together at the metaphase plate. This is different from mitosis and meiosis 2 because they're pairing up at the plate, not lining up individually as chromosomes. And then lastly, in anaphase 1, because the pairs of homologs lined up together, that's what's going to be separated during anaphase. So in meiosis 1, anaphase 1, we separate the homologous chromosomes, unlike in mitosis and meiosis 1, where we separate the sister chromatids.